Good morning, everyone, and a warm welcome to today's session. I'm delighted to have you here. Today, we'll be exploring good practices for microbiology laboratories, and this is just the first part of our series. This presentation will cover essential guidelines to ensure quality and safety in microbiology labs. So, let's get started and look forward to an engaging and informative session. Let's start by talking about personnel, environment, and premises. These are the foundation of a reliable pharmaceutical microbiology laboratory. Having well-trained staff, a controlled environment, and properly maintained premises are essential for ensuring accurate and consistent results. Next, I want to emphasize the importance of environmental monitoring in the laboratory. Regular monitoring helps us detect any potential contaminants early and maintain a safe, sterile environment. This is critical for maintaining the integrity of our tests and ensuring product safety. Moving on to cleaning, disinfection, and hygiene. These practices are vital to prevent cross-contamination and microbial growth. Consistent cleaning protocols and hygiene practices safeguard the quality of our work and help us meet regulatory standards. Now, let's consider sterility test facilities. These are specially designed areas where we conduct tests to confirm that products are free of microbial contamination. Proper design and control of these facilities are key to obtaining valid results. Validation of test methods is another critical point. We need to ensure that our methods are accurate, reliable, and reproducible. Validated methods form the backbone of compliant and trustworthy testing. Turning to equipment, its maintenance, qualification, calibration, and performance verification are essential steps. Regular maintenance keeps equipment functioning optimally, while qualification and calibration ensure that our instruments produce precise, accurate results. Finally, I want to highlight that all of these elements come together within pharmaceutical microbiology laboratories. They work in harmony to support quality control, ensuring that our pharmaceutical products meet the highest standards for safety and efficacy. Let's take a closer look at the core components of pharmaceutical microbiology laboratories. First, we have reagents and culture media, which are essential for growing and identifying microorganisms. Reagents are the chemicals used in various testing procedures, while media provide the nutrients needed for organisms to grow. Next, proper labeling is crucial to ensure that all samples and materials are correctly identified and traceable throughout the testing process. This helps prevent mix-ups and maintains accuracy. Organism resuscitation is another key point. It's about reviving microorganisms from storage so they can be accurately tested and identified. Using reference materials and reference cultures ensures that our testing is reliable, as they serve as standards for comparison. International standards and pharmacopoeial reference substances guide us in maintaining consistency and quality across different laboratories and regions. Reference cultures are used for calibration and validation of our microbiological methods. Sampling is a fundamental step, collecting representative samples carefully to ensure valid results. Once samples are collected, handling and identification are critical to prevent contamination and to accurately determine the presence or absence of microorganisms. All these elements are interconnected and play a vital role in ensuring the safety, efficacy, and quality of pharmaceutical products. They form the backbone of microbiological testing in our laboratories, aligning with our overall commitment to high standards. Now, let's look at some essential aspects of pharmaceutical microbiology laboratories. First, the disposal of contaminated waste is critical to prevent any cross-contamination or environmental issues. Proper procedures must be followed to ensure safety and compliance. Next, the quality assurance of results and quality control of performance are fundamental to maintaining accuracy and reliability in our testing processes. This involves implementing internal quality control measures, which help us monitor and verify that our testing procedures are consistent and trustworthy. Speaking of testing procedures, they need to be well-defined and rigorously followed to ensure valid results. Lastly, Test reports are the final product of our efforts. They must be clear, accurate, and comprehensive, serving as a solid basis for decision-making in pharmaceutical quality management. All these elements are interconnected and form the backbone of a robust microbiology laboratory, ensuring that our products are safe and effective for consumers. In this slide, we're focusing on the pharmaceutical microbiology laboratories, and I want to start by emphasizing the scope of what these labs do. They play a crucial role in ensuring the safety and quality of pharmaceutical products. Some of the main activities include sterility testing, which is essential for confirming that products are free from viable microorganisms. Additionally, these laboratories are responsible for detecting, isolating, enumerating, and identifying different microorganisms such as bacteria, yeasts, and molds. They also test for bacterial endotoxins in various materials, including starting materials and water, as well as in the final products, surfaces, garments, and the environment. This comprehensive testing helps us understand contamination levels and microbial presence across the manufacturing process. Another key activity is performing assays that use microorganisms as part of the test system, which supports various quality control processes. It's important to note that the World Health Organization provides guidelines covering these activities, offering internationally recognized recommendations to ensure consistency and reliability. Moreover, these guidelines are complemented by other standards and pharmacopoeia references, which collectively form the foundation for good microbiological practices in the pharmaceutical industry. Connecting this to the overall theme, these microbiology activities are vital for safeguarding public health by ensuring that pharmaceutical products meet strict safety criteria throughout their life cycle. Let's take a closer look at the personnel involved in pharmaceutical microbiology laboratories. These laboratories rely heavily on skilled professionals who are trained specifically to handle microbiological testing and ensure the quality and safety of pharmaceutical products. Their expertise is crucial because they are responsible for detecting and controlling microbial contamination, which directly impacts patient safety. The personnel typically include microbiologists, laboratory technicians, and quality assurance staff. Microbiologists are the experts who design and oversee testing procedures, interpret results, and troubleshoot any issues that arise. Laboratory technicians support these efforts by performing routine testing and maintaining the lab environment. Quality assurance personnel ensure that all procedures adhere to regulatory standards and good manufacturing practices. Connecting this to the overall theme, the competence and diligence of the laboratory personnel are fundamental to maintaining the integrity of pharmaceutical products. Their role is vital in safeguarding public health by ensuring that only safe, contaminant-free medicines reach the market. Now, let's take a look at the personnel in pharmaceutical microbiology laboratories. It's essential that testing and supervision are carried out by experienced individuals who are qualified in microbiology or a similar field. This ensures that the work is reliable and compliant with industry standards. Training plays a crucial role here. Personnel should have both formal education and relevant practical experience to perform their tasks effectively. It's also important that current job descriptions are maintained for everyone involved in testing, calibration, validation, or verification activities. This helps clarify roles and responsibilities, ensuring accountability. Additionally, keeping detailed records of all technical personnel, including their qualifications, training, and experience, is vital. These records provide a clear overview of the team's expertise and help demonstrate competence during audits or inspections. Finally, when it comes to interpreting test results, only authorized personnel with suitable experience and relevant knowledge should make opinions or interpretations. Their expertise is key to ensuring that results are accurately understood and appropriately acted upon. All of these points collectively support the integrity and reliability of our microbiological testing processes, which is at the core of maintaining high-quality pharmaceutical products. Let's talk about the importance of training in pharmaceutical microbiology laboratories. First, All personnel must have adequate training, which covers a range of essential skills. This includes performing various tests accurately and operating laboratory equipment safely and efficiently. It's also crucial that staff are trained in basic techniques such as plate pouring, counting colonies, and maintaining aseptic conditions. These fundamental skills ensure reliable results and prevent contamination. Additionally, media preparation, serial dilutions, and identification techniques are vital for accurate microbiological analysis. Beyond technical skills, personnel need to be trained in containment procedures to prevent microorganisms from escaping the laboratory environment. Safe handling of microorganisms is equally important to protect both staff and the environment. To maintain high standards, competence is continuously monitored. If someone needs it, retraining is provided to keep skills sharp and ensure compliance with safety and quality protocols. By investing in thorough training and ongoing monitoring, we ensure that everyone in the lab contributes to a safe, effective, and compliant microbiological testing process. This is fundamental to delivering safe pharmaceutical products and maintaining regulatory standards. Now, let's talk about pharmaceutical microbiology laboratories. It's crucial to remember that access to these laboratories should be strictly restricted. This is to ensure the integrity of the work and to prevent any contamination or accidental exposure. Personnel working in these areas need to be fully aware of the proper procedures for entering and exiting. This includes gowning protocols, which are essential to maintain a sterile environment. Knowing the intended use of each specific area helps in understanding why certain precautions are necessary. It's also important to recognize that there are restrictions on working within these areas. These restrictions are in place for safety reasons and to protect the quality of microbiological testing. Understanding the reasons behind these restrictions helps reinforce compliance and awareness. Lastly, personnel should always be conscious of the appropriate containment levels required for different tasks. This ensures that the laboratory maintains the necessary biosafety standards to protect staff, the product, and the environment. All these points are vital to maintaining a controlled, safe, and compliant microbiology laboratory environment. When it comes to pharmaceutical microbiology laboratories, one of the most critical aspects is having dedicated and separated areas along with support equipment. For example, autoclaves and glassware should be designated specifically for certain tasks to prevent cross-contamination. The design of these labs must be appropriate for the operations we conduct. This means having enough space to work efficiently and safely. Proper layout helps us avoid mix-ups, contamination, and cross-contamination, which are vital for maintaining the integrity of our microbiological processes. Using suitable materials of construction is also essential. These materials should allow for effective cleaning and disinfection, which helps minimize contamination risks. Think of it as creating an environment that's easy to keep sterile and controlled. Lastly, we need enough space for everything: samples, reference organisms, media, sometimes even with cooling if necessary, testing, and records. And it's important to have separate storage areas for certain media and samples to prevent any accidental mix-ups or contamination. All these points come together to ensure our microbiology labs operate smoothly, safely, and in compliance with good manufacturing practices, which is at the heart of quality control in pharmaceuticals. Let's take a closer look at pharmaceutical microbiology laboratories. One of the key aspects is ensuring a separate air supply for both the laboratories and the production areas. This separation helps maintain a controlled environment, particularly regarding temperature and humidity, which are crucial for microbial control. It's also vital that the air quality in these spaces is appropriate. It shouldn't be a source of contamination. That means implementing proper filtration and air handling systems to keep the environment clean and safe. Within these labs, we need segregated and classified areas for specific tasks like sample preparation, media and equipment preparation, and the enumeration of microorganisms. By clearly defining these zones, We reduce cross-contamination risks and improve process control. Of course, not all areas need to be dedicated solely to microbiology. Based on risk management principles, non-dedicated areas can be used, but only if proper controls are in place. This flexibility allows for efficient use of space while still maintaining the integrity of our microbiological processes. All these measures together form a comprehensive approach to ensuring that our microbiology laboratories operate under optimal conditions, supporting the overall quality and safety of pharmaceutical products. Let's talk about pharmaceutical microbiology laboratories. Firstly, it's important to understand that classification should be based on the criticality of the work being done. This means that areas handling more sensitive or critical processes require stricter controls and classifications. Next, sterility testing is a key part of ensuring product safety, and it should always be performed in a dedicated area. This helps prevent any contamination from other activities or environments that could compromise the test results. Furthermore, equipment used in microbiology labs should not be moved between areas of different cleanliness classes. And if equipment needs to be used outside the microbiology area, there should be specific precautions in place to prevent cross-contamination. This is crucial because cross-contamination can jeopardize both the integrity of the testing process and the safety of the products. Overall, these practices help maintain a controlled environment, ensuring reliable results and product safety throughout the manufacturing process. Now, let's delve into the importance of environmental monitoring in pharmaceutical microbiology laboratories. This is a critical component to ensure the sterility and safety of our products. An environmental monitoring program should be implemented wherever necessary, particularly in areas dedicated to sterility testing. We use various methods to monitor the environment, such as active air monitoring, which involves sampling the air actively to detect any microbial presence. Additionally, we utilize air settling plates or contact plates to assess surface contamination. Monitoring temperature and pressure differentials also helps us maintain the optimal environment and quickly identify any deviations that could impact product quality. It's essential to set alert and action limits for these monitoring results. These thresholds help us determine when results are within acceptable ranges or if immediate action is required. Trending these results over time allows us to identify patterns or potential issues before they become serious problems. Cleaning, disinfection, and maintaining hygiene are fundamental to controlling microbial contamination. We have a detailed cleaning and disinfection program and regularly review the results to ensure effectiveness. In case of spillages, having a standard operating procedure, or SOP, is vital. This guides us on how to respond quickly and effectively to minimize contamination risks. Finally, proper hand hygiene facilities, such as hand washing and hand disinfection stations, are crucial. They play a key role in preventing the transfer of microbes into the controlled environment, safeguarding the integrity of our products and the safety of our patients. Let's now look at the pharmaceutical microbiology laboratories, specifically the sterility test facilities. These areas have very strict environmental requirements, including cleanroom standards, to ensure the integrity of the testing process. It's crucial that all procedures are performed under aseptic conditions to prevent contamination. Typically, we operate in a Class A environment, which is equivalent to ISO 4.8, within a Class B background. The Class B background provides a controlled environment with unidirectional airflow, which is essential for maintaining asepsis. Alternatively, these tests can be conducted within barrier isolators or biosafety cabinets that also provide a protective environment. The design of the facility layout and room airflow patterns plays a vital role. It's important that these airflow patterns are carefully planned and maintained so that they are not disrupted, which could compromise the sterile environment. Proper airflow helps ensure that any contaminants are swept away and do not reach the critical testing areas. Finally, all premises, services, and equipment involved in these processes must undergo qualification, meaning they are tested and verified to meet the required standards. This qualification process helps guarantee that every aspect of the environment and equipment supports a sterile, contamination-free testing process, aligning with our overall goal of ensuring product safety and quality. Now, let's turn our attention to pharmaceutical microbiology laboratories. These labs are a critical component of the overall quality control process in pharmaceutical manufacturing. They are specifically dedicated to testing and monitoring the microbiological quality of raw materials, active ingredients, and finished products. The goal here is to ensure that the products are safe, effective, and free from harmful microbial contamination. Microbiology laboratories play a crucial role in identifying microorganisms, assessing microbial limits, and evaluating sterilization processes. They help us detect any potential microbial risks early on, which is vital for maintaining product integrity and patient safety. Throughout the presentation, we've emphasized the importance of rigorous testing and compliance with regulatory standards. These laboratories are the frontline in safeguarding public health by ensuring that pharmaceutical products meet the highest microbiological quality standards. In essence, pharmaceutical microbiology laboratories are the guardians of microbial quality, supporting the overall theme of quality assurance in pharmaceutical manufacturing. When it comes to pharmaceutical microbiology laboratories, maintaining a high level of cleanliness and proper air handling is absolutely essential. That's why the cleanroom classification and the air handling equipment in our sterility test facilities should be requalified at least once a year. Of course, if there's no significant risk, the frequency can be adjusted, but regular requalification is key. During these requalification tests, we typically check non-viable and viable limits to ensure the environment remains within acceptable microbial limits. We also verify the integrity of HEPA filters, which are critical for maintaining air purity. Additionally, room airflows need to be assessed to confirm that air is moving correctly and preventing contamination. Another important aspect is having clear SOPs, standard operating procedures, that specify where sample points are mapped out for routine monitoring. These SOPs should detail the exposure duration and the frequency of all microbiological environmental monitoring activities. This helps ensure consistency and thoroughness in our testing. And of course, personnel play a vital role. Operators should be appropriately dressed in garments designed for clean environments, and they need to be trained and certified in gowning procedures. Proper gowning minimizes contamination risks and keeps our sterile environments safe. All of these measures are interconnected and essential for maintaining the integrity of our sterility testing process, ultimately ensuring the safety and efficacy of the pharmaceutical products we produce. Now, let's take a closer look at the key features of pharmaceutical microbiology laboratories. First, the grade A and B zones are critical areas that are supplied with terminal HEPA filters. These filters are essential because they ensure that the air within these zones remains as free from contaminants as possible, maintaining the high standards required for pharmaceutical work. Next, we have airflow alarms and pressure differentials, along with indication instruments. These tools serve as the eyes and ears of the cleanroom environment, alerting us immediately if there's any deviation from the set parameters, such as a drop in pressure or compromised airflow. Monitoring these aspects helps us maintain a consistently controlled environment. Room pressure readings are taken regularly and recorded. This practice is vital because it provides a documented history of the environment's stability, making it easier to identify potential issues before they impact the manufacturing process. It's also important that pressure gauges are clearly labeled to indicate which area they serve and the acceptable pressure specifications. Clear labeling helps prevent confusion and ensures quick responses when adjustments are needed. Entry to these clean rooms is controlled via airlocks of suitable size. This design minimizes contamination by controlling airflow and restricting unnecessary access, which is especially important in pharmaceutical microbiology. Finally, the final change room should be kept under at-rest conditions, meaning no activity occurs there, and should be of the same grade as the room it serves. This consistency helps maintain environmental integrity right up to the point of product handling or testing. Overall, these measures work together to create a controlled, sterile environment essential for pharmaceutical microbiology, ensuring product safety and quality at every step. When it comes to pharmaceutical microbiology laboratories, environmental monitoring is a critical component to ensure product quality and safety. It's important that the monitoring methods we choose reflect the specific facility we're working in, whether that's a regular room or an isolator. We need to use a combination of air and surface sampling techniques that are appropriate for the environment. For air sampling, active air sampling methods are common, where we actively draw air through a device to capture microorganisms. Settle plates, also known as exposure plates, are another useful tool. They allow particles to settle naturally onto a growth medium over time, giving us a snapshot of airborne contamination. Surface sampling is equally important, and we often use contact plates, swabs, or flexible films to detect microbial presence on surfaces. Replicate organism detection and counting plates, or RODAC plates, are specially designed for this purpose. They let us directly sample surfaces and quantify the microbial load. Additionally, operator glove prints are a valuable monitoring method, especially during aseptic processing, because they help us assess the cleanliness of gloves that come into contact with the product or environment. Overall, the key is selecting the right combination of these methods based on the specific area we're monitoring. By doing so, we can maintain a controlled environment, minimize contamination risks, and uphold the highest quality standards in pharmaceutical manufacturing. Let's talk about pharmaceutical microbiology laboratories and their role in ensuring product safety. A key part of this is microbial environmental monitoring of the sterility test zone. This monitoring isn't just a one-time check. It happens during every work session. That way, we can catch any contamination risks in real time. Importantly, these tests are conducted under operational, or dynamic, conditions, meaning we're monitoring the environment exactly as it functions during actual production. To guide us in this process, we have written specifications that clearly define what is acceptable. These include alert and action limits for microbial contamination. Alert limits indicate when we need to be cautious and investigate further, while action limits tell us when immediate corrective actions are necessary. All of these measures are designed to maintain a sterile environment and ensure the integrity of our products. Monitoring consistently and setting clear limits are vital to managing contamination risks effectively, which ties back to our overall goal of quality assurance in pharmaceutical manufacturing. When we talk about the validation of test methods, it's crucial to understand the different categories involved. For compendial methods, the key requirement is demonstrating that these methods are suitable for recovering bacteria, yeast, and mold, even in the presence of the specific product we're testing. This means we need to verify the method itself, making sure it works reliably, and also confirm the test's suitability. This involves using positive controls to show the method can detect what it's supposed to, and negative controls to ensure there's no false positive. Moving on to non-compendial or other recognized reference methods, these need to be validated before we use them. Validation here covers aspects like accuracy, which tells us how close our measurements are to the true value, precision, or how reproducible our results are, and specificity, ensuring the method detects only the target organisms. We also look at limits of detection and quantitation, which define how sensitive our methods are, as well as linearity, to confirm the method's response is proportional to the concentration, and robustness, to see if the method can withstand small variations without losing accuracy. An important consideration is whether the product might inhibit the test. So, potential inhibitory effects must be carefully evaluated because they can lead to false negatives or inaccurate results. And to ensure our data is reliable, we employ appropriate statistical methods, These are often guided by pharmacopoeias to analyze our results correctly. All of these points underscore the importance of rigorous validation in pharmaceutical microbiology laboratories. Without proper validation, our microbial testing might not be trustworthy, which is why these standards are essential for maintaining quality and safety throughout the product lifecycle. When it comes to equipment in pharmaceutical microbiology laboratories, it's crucial that each item, whether it's an instrument or any other device used for testing, verification, or calibration, is uniquely identified. This helps us track and manage each piece effectively, ensuring accountability and traceability. We also need to have a documented program in place that covers all aspects of equipment management. This includes qualification, calibration, performance verification, and maintenance. Having a clear, written plan ensures that every step is followed consistently and that the equipment remains reliable and accurate over time. Monitoring the use of equipment is another key point. We should have systems in place to oversee how and when equipment is used, which helps prevent misuse or oversight. Additionally, having standard operating procedures, or SOPs, for maintenance is vital. These SOPs should specify the necessary maintenance activities at predetermined intervals, ensuring the equipment remains in optimal condition. All these activities should be thoroughly recorded. Keeping detailed records not only supports compliance, but also makes audits smoother and helps us troubleshoot if issues arise. In the context of pharmaceutical microbiology laboratories, these practices are fundamental. They help maintain the integrity of our testing processes, ensure data accuracy, and ultimately protect product quality and patient safety. So overall, effective equipment management is a cornerstone of a compliant, reliable microbiology lab. It ties into the larger theme of quality assurance and regulatory compliance that runs throughout our entire operation. Let's take a moment to talk about qualification, which is a crucial aspect when it comes to equipment in pharmaceutical microbiology laboratories. For a more detailed understanding, I recommend reviewing the training modules on good practices for pharmaceutical quality control laboratories. These modules cover the essential steps involved in qualifying equipment. First, we have design qualification, or DQ, which ensures that the equipment design meets the specific requirements of your laboratory operations. Next is installation qualification, IQ, where we verify that the equipment has been installed correctly and according to specifications. Following that is operational qualification, OQ, which tests whether the equipment functions properly within defined parameters. And finally, performance qualification, PQ, confirms that the equipment consistently performs effectively under real-world conditions. All these steps are vital to maintaining the integrity and reliability of our microbiology testing processes. Remember, proper qualification not only ensures compliance, but also directly impacts the quality of the pharmaceutical products we produce. So, understanding and implementing these qualification stages thoroughly is key to maintaining high standards in our laboratories. Let's talk about calibration, performance verification, and monitoring of use. First, you'll notice that instruments are labeled with dates. This helps us keep track of when each piece of equipment was calibrated or verified. The key here is determining the right frequency for these activities. We need to calibrate and verify performance often enough to catch any drift before it causes issues. The intervals between calibration and verification should be shorter than the time it takes for equipment to drift outside acceptable limits. This way, we ensure that our instruments remain accurate and reliable during their use. Remember, equipment must perform according to predefined acceptance criteria, meaning they should meet specific standards to be considered functioning correctly. In pharmaceutical microbiology laboratories, maintaining this rigorous schedule is crucial. It helps us uphold the integrity of our testing processes and ensures consistent, trustworthy results. Overall, the goal is to keep our instruments in optimal condition so that our laboratory performance remains precise and compliant with regulatory expectations. Now, let's look at some examples of equipment maintenance. The upcoming slides will provide specific cases to help illustrate these points. It's important to remember that the frequency of maintenance should be determined by the actual needs of the equipment, its type, and how it has performed previously. Additionally, always refer to the manufacturer's manuals for their specific recommendations. In pharmaceutical microbiology laboratories, maintaining equipment properly is crucial to ensure reliable results and a sterile environment. By tailoring maintenance schedules to the equipment's performance and following supplier guidelines, we can prevent issues before they arise and keep our laboratory running smoothly. Let's take a closer look at pharmaceutical microbiology laboratories. These labs are a critical component in ensuring the safety and efficacy of pharmaceutical products. They are specialized facilities where we carry out microbiological testing to detect any microbial contamination in drugs, raw materials, and the manufacturing environment. The key points here are the importance of maintaining strict aseptic conditions and adhering to regulatory standards. These labs are equipped with advanced technologies and follow rigorous protocols to identify bacteria, fungi, and other microorganisms that could compromise product quality. Connecting this to our overall theme, these labs serve as the frontline defense in protecting public health. They help us ensure that every batch of medication meets the highest safety standards before it reaches patients. This way, we can prevent potential contamination issues and uphold the integrity of pharmaceutical products. Let's now turn our attention to pharmaceutical microbiology laboratories. These laboratories play a crucial role in ensuring the safety and quality of pharmaceutical products. They are specialized facilities where microbiological testing is conducted to detect and control microbial contamination. This is essential because even the smallest microbial presence can compromise drug safety or efficacy. In these labs, we perform a variety of tests, such as sterility testing, endotoxin testing, and microbial limit testing. Each of these is designed to identify specific microbial threats and ensure compliance with regulatory standards. It's also important to highlight that these laboratories are built and operated under strict Good Laboratory Practices, or GLP, to maintain accuracy and reliability of results. They use sophisticated equipment and follow validated methods to ensure consistency. Overall, pharmaceutical microbiology laboratories are a cornerstone in the manufacturing process, helping us uphold the highest standards of safety and quality in our medicines. Their work supports the entire pharmaceutical quality system and protects patient health. Let's take a moment to focus on pharmaceutical microbiology laboratories. These labs are a crucial part of the overall quality assurance process in pharmaceutical manufacturing. They are responsible for testing and monitoring microbial contamination, ensuring that products are safe and effective for patient use. In these laboratories, we conduct a variety of microbiological tests, from detecting bacteria and fungi to assessing endotoxin levels. These tests help us verify that manufacturing environments, raw materials, and finished products meet strict regulatory standards. The key point here is that microbiology labs act as the frontline defense against microbial contamination. They help us identify potential issues early and maintain the integrity of pharmaceutical products. Connecting this to our overall theme, maintaining rigorous microbiological control is essential for patient safety and product efficacy. It's not just about compliance, it's about protecting health and ensuring trust in our medicines. Let's take a look at some examples of equipment qualification and monitoring. Keep in mind, the specific details I'm sharing are just examples. The actual frequency of these activities will depend on several factors. These include the needs of the equipment, the type of equipment, its previous performance, and how critical it is to the process. In a pharmaceutical microbiology laboratory, for instance, equipment plays a crucial role in ensuring the quality and safety of our products. That's why it's essential to establish a clear schedule for qualification and ongoing monitoring. This helps us catch potential issues early and keeps everything within the required specifications. So, the key takeaway here is that our approach to qualification and monitoring should be tailored to each piece of equipment based on these various factors to maintain the integrity of our processes and products. Let's take a closer look at pharmaceutical microbiology laboratories. These labs play a crucial role in ensuring the safety and quality of pharmaceutical products. They are responsible for detecting and controlling microbial contamination throughout the manufacturing process. By doing so, they help prevent the release of products that could be harmful to patients. In these labs, we employ a variety of microbiological testing methods, from identifying bacteria and fungi to testing for endotoxins. The goal is to maintain strict sterility and uphold good manufacturing practices. It's important to recognize that the work done here directly ties into our overall commitment to product integrity and patient safety. These laboratories are the frontline defenders against microbial contamination, ensuring that everything produced meets the highest standards. So, understanding how pharmaceutical microbiology laboratories operate is key to appreciating the robustness of our quality control systems and the safety of the medicines we rely on. Let's take a closer look at pharmaceutical microbiology laboratories. These labs play a crucial role in ensuring the safety and quality of pharmaceutical products. They are specialized facilities dedicated to testing for microbial contamination, which is vital for safeguarding patient health. In these laboratories, we perform various microbiological tests to detect bacteria, fungi, and other microorganisms that could compromise drug safety. It's important to highlight that these labs adhere to strict regulatory standards and good laboratory practices, or GLP, to maintain accuracy and reliability of results. They also implement rigorous sterilization and contamination control procedures to prevent any cross-contamination. Overall, pharmaceutical microbiology labs are a cornerstone of quality assurance in the industry. They help us verify that our products meet safety standards before reaching patients, which ties directly into our overarching goal of ensuring drug efficacy and safety. Let's take a closer look at pharmaceutical microbiology laboratories. These labs are essential components of the pharmaceutical industry, ensuring the safety and quality of the products we rely on. They focus on detecting and controlling microbial contamination in drugs, raw materials, and manufacturing environments. The key points here are the meticulous testing procedures and strict quality control standards that must be maintained. These labs use specialized techniques to identify bacteria, fungi, and other microorganisms that could compromise product safety. It's important to understand that microbiology labs play a critical role in compliance with regulatory requirements, such as those from the FDA or EMA. They help ensure that every batch of medication meets the highest safety standards before reaching patients. Connecting this back to our overall theme, these laboratories exemplify the rigorous scientific processes behind pharmaceutical quality assurance. They are vital in safeguarding public health by preventing microbial contamination and ensuring product integrity throughout the manufacturing process. When it comes to temperature measurement devices, it's crucial to understand their role in ensuring accurate results. Since temperature can directly influence the outcome of an analysis or is vital for the proper functioning of equipment, using the right quality tools is essential. For example, in pharmaceutical microbiology labs, we often rely on devices like liquid-in-glass thermometers, thermocouples, and platinum resistance thermometers, also known as PRTs, especially when working with incubators and autoclaves. These devices must be of appropriate quality to meet the accuracy requirements of your specific process. Remember, calibration is just as important. It should be traceable to recognized national or international standards. This traceability guarantees that your measurements are consistent and reliable, which is fundamental in our field. Overall, maintaining properly calibrated, high-quality temperature measurement devices ensures the integrity of your results and supports compliance with regulatory standards. It's a key part of good laboratory practice and critical for producing trustworthy microbiological data. When working with incubators, water baths, and ovens, It's crucial to understand that their temperature stability and distribution play a key role in ensuring reliable results. Initially, you should establish and document how stable the temperature is, how evenly it's distributed throughout the equipment, and how long it takes to reach equilibrium. This is especially important for typical uses, like stacking Petri dishes, so factors such as their position, spacing, and height should be considered and recorded. It's also essential to perform these checks during the initial validation process and to keep records after any significant repairs or modifications. This helps ensure that the equipment continues to perform as expected. Regularly monitoring the operating temperature is vital, and all records of these checks should be retained for compliance and traceability. In pharmaceutical microbiology laboratories, maintaining precise and consistent temperature conditions is fundamental. It guarantees the integrity of your experiments and the accuracy of your results. Let's talk about autoclaves, including media preparators. These are essential tools in ensuring sterilization processes are effective and consistent. It's crucial that autoclaves meet specific time, temperature, and pressure tolerances to guarantee reliability. To achieve this, we use calibrated sensors and verified timers, which help us monitor and control the sterilization cycle precisely. Validation plays a key role here. It's not just about initial setup, but ongoing verification. This includes conducting temperature distribution studies for each operating cycle and load configuration, ensuring uniform heat distribution. We also perform heat penetration studies to confirm that sterilization reaches all parts of the load effectively. When it comes to media preparators, we typically use two sensors to get a more accurate picture of the sterilization process. Remember, validation isn't a one-time task. Revalidation should consider all phases, heating, sterilization, and cooling, to maintain confidence in the process over time. This whole approach is vital for our pharmaceutical microbiology laboratories. It ensures our sterilization processes are robust, consistent, and compliant with regulatory standards, ultimately safeguarding product quality and patient safety. Let's talk about some essential aspects of maintaining our autoclave processes. First, it's crucial to have clear operating instructions, or SOPs, that everyone follows to ensure consistency and safety. These SOPs should outline acceptance and rejection criteria, so we know exactly what standards need to be met for a cycle to be considered successful. We also need to keep detailed records of every autoclave cycle, including temperature and time. This documentation is vital for traceability and quality assurance. Monitoring how well the autoclave performs can be done in a couple of ways. One method is using a thermocouple and recorder to produce a chart or printout, giving us a visual confirmation of the cycle parameters. Alternatively, direct observation can be used to record the maximum temperature achieved and how long it was maintained. In addition, chemical or biological indicators, as well as autoclave tape, serve as important tools to verify sterilization effectiveness. And in some cases, especially when dealing with highly contaminated materials, a separate autoclave for decontamination is recommended to prevent cross-contamination. Finally, in our pharmaceutical microbiology laboratories, these practices are part of a broader quality control framework, ensuring that every step of sterilization is properly documented, monitored, and validated to maintain the highest standards of safety and efficacy. Let's talk about weights and balances. It's essential to perform regular calibration to ensure accuracy, and this calibration must be traceable to standard weights and certified standard weights. This helps us maintain consistency and confidence in our measurements. When it comes to volumetric equipment, initial verification is crucial. This applies to automatic dispensers, dispenser and diluter systems, mechanical hand pipettes, and disposable pipettes. After the initial check, regular assessments are necessary to ensure ongoing precision. We verify the accuracy of the volume delivered by these instruments by comparing it against the set volume, especially important for instruments that offer multiple volume settings. We also check their precision, including single-use pipettes, to guarantee consistent performance. All these steps are vital in our pharmaceutical microbiology laboratories because they underpin the reliability of our results and help us adhere to strict quality standards. Consistent calibration and verification are the backbone of maintaining the integrity of our testing processes. When it comes to other equipment in the pharmaceutical microbiology lab, it's really important to ensure that all instruments are properly verified regularly. For example, conductivity meters, oxygen meters, pH meters, and similar tools should be checked either on a routine schedule or just before each use. This helps guarantee the accuracy of your measurements every time. Buffers used in testing need to be stored properly and must be within their expiry date. Using expired buffers can compromise your results, so always double-check their storage conditions and expiry dates. If you're working with hygrometers, calibration is key. These need to be calibrated regularly to ensure humidity readings are accurate, which is vital in maintaining proper environmental controls. Timers are another critical piece of equipment. They should be verified with a calibrated timer or synchronized with a national time signal to ensure timing precision during experiments. For centrifuges, it's essential to verify their rotations per minute, or RPM. Especially when precise centrifugation is crucial for your process or testing, the centrifuge should be calibrated to confirm it's running at the correct speed. All these practices, regular verification, calibration, and proper storage, are fundamental to maintaining the integrity of your microbiology testing processes and ensuring reliable, reproducible results in your lab. For further support, please contact us. We're here to help with any questions or additional information you might need. Thank you so much for your time and attention throughout this presentation. Remember, we're committed to supporting you every step of the way, so don't hesitate to reach out if you need anything.